Hello, my name is Tosin Taiwo. I'm a senior clinical scientist at the Clinical Genomics department, and today I'm going to be talking you through how a sample comes into our lab for investigations from central specimen reception all the way through to data analysis. So we've just had a batch of samples delivered, and now they're going to go into the central specimen reception. Follow me. So here's our central specimen reception. This is where all the samples come in through to specialist pathology for acceptance, and our staff go through each request and determine what testing is required and what laboratory to take you to. I've got here Henry with me, who's one of our very experienced member of staff, and is the specimen reception manager. Is going to go through the process with us. Henry?
Hi there. Yeah, so we receive samples from external referrers, from the GLH, from referrers for our hematological malignancy diagnostic service, and other internal samples from the hospital. Our staff will receive these. Many of these are raised on our electronic order com system, and we will receive these. Our staff will identify what samples we have and which labs they need to go to. So we receive samples for the genomics labs for molecular and cytogenetics, and also for our flow lab and our histology. We get a mixture of, as I say, hemoc samples, we get solid tumour samples, and these are all registered on our EPIC system, the documentation scanned in, and then they are labelled up and put for collection by the laboratories, ready to go on to the next stage.
Thank you, Henry. That was very informative. So now that the samples have been accepted into specimen reception, the samples that require genomics testing are then taken into the clinical genomics laboratory. So the next step is to process the samples, and I'm going to show you the instruments that we use and the members of staff that are involved in that side of the process. This is specimen extraction. This is where the first step of the sample processing occurs. So I've got here with me today Oli. He's one of our trainee clinical scientists, and he's going to tell us what happens next. Oli?
So the first thing we need to do for extraction is extract the DNA or RNA from the sample. This is where all the genetic information is stored for our analysis. So for a blood sample, we've got specific collection tubes which contain preservatives which prevent the sample from degrading. This sample, we either have blood samples or bone marrow samples which are analysed for blood cancers. Then moving on, we've got our solid tumour samples. These samples are fixed to microscope slides, to prevent the sample from degrading again. And for these samples, we have a specific marked out H&E slide, and the H&E slide will show where the tumour is located within the tissue sample, which means that the tumour DNA can be extracted and we can get the highest yield of tumour DNA. This used to be quite a manual process, but now it's really exciting that we have the automator machine, which means that the process can be much quicker.
Thank you very much. Okay, let's find somebody to tell us about this automator. Sounds very interesting. Come with me. Frankie.
Hi.
Hi. So Frankie's one of our genetic technologists, and she's going to tell us about automata.
So I've already mentioned this is the automata system. He also mentioned that it used to be a very manual process, extracting DNA and RNA. Basically, automata just automates a lot of the manual steps that used to take us technologists a lot of hours. We can now, it's a very high throughput process, so we can process a lot of samples much quicker. At the moment, we're running saliva and blood and bone marrow samples in a combined run. Automata is really great as it speeds up the whole process, which allows patients to get their results quicker. It's very high throughput, as I mentioned, and it just reduces errors in a lot of the steps are now done on the system, and therefore reduces human error.
Thank you very much. And I presume it would allow patients to get their results much quicker as well, because we're doing so many samples at the same time. Thanks very much, Frankie. That sounds great. Okay, so once DNA and RNA extraction have been completed, then the next step is to process the sample for the actual test that's coming through, which is next generation sequencing. So I'm going to try and find one of our scientists to talk to us about that aspect of things. The steps that it takes to get to that stage. Oh, hi Lizzie. How are you doing today?
Doing a library prep for NGS.
Okay.
So to be able to prepare DNA and RNA samples, to be able to be sequenced, we need to do a couple of steps. So first we need to break the DNA down into smaller pieces and add special adapters or barcodes so that when we sequence them, we can tell the patients apart. This process can take around a day and can be quite labor intensive. So now we have moved on to automating this process on our Hamilton machines, which allow the process to be done without lots of manual steps, and it makes it much more quicker and more efficient to be able to do that.
Thank you very much. So how many samples roughly do you process at the same time?
So we can do up to 96 samples at the same time, and we have multiple machines, so we could run separate runs.
Thank you, Lizzie. So Lizzie's one of our trainee clinical scientists helping us in the lab today. Thanks, Lizzie. Okay, so once library preparation is completed, now it's time to sequence. So come along with me. Let's see if we can find somebody to tell us about sequence. So I've changed my lab coat because now we're going into a different zone from where we were before. Previously, we were in a place called pre-PCR, and now we're going into post-PCR, where sequencing occurs. Come inside. Hello. Oh, hi. So this is Paula, one of our very, very experienced genomic scientists and translational research laboratory manager. Paula, tell us about NGS sequencing.
Hello. So you've heard how the DNAs are made into sequencing libraries, and then we need to load them onto the sequencing instrument. Essentially, what that machine does is it's a big camera that takes pictures of all the four bases in the DNA, A, B, G, and C, from all the samples that are pulled together and captured for the regions of interest that are parts of the DNA that are involved in cancer pathways and we want to have a look and explore. That data can then be analyzed through our very complex bioinformatics pipeline and gives information about cancer pathways involved. We can also get information about treatment that would be matched to the mutations found in the cancer samples, give us predictive biomarkers or any other information that we may be interested in. And these instruments have the highest capacity, and we also have the N6, which are used for a bit less samples. This is NovaSeq flow cell, to which the samples are loaded. And this is the highest capacity flow cell used for the NovaSeq X. So each of those machines have different throughputs, so we load different amounts of samples, different amounts of patient samples on, just to keep our turnaround times, so reagent and record delivery as short as possible for the patients.
Thank you so much, Paula. So when we heard about library prep earlier from Lizzie, she mentioned that we tend to process about 96 samples at the same time. So, do you all like to see samples all at the same time?
Actually, even more. So we
Wow.
have the capacity to sequence 384 samples on one close on this instrument. So 384 patients all together are analyzed in this sequence.
That sounds really, really cool.
Thank you. Thanks a lot.
So, sequencing marks the end of the wet laboratory process. Once sequencing has been performed, as nicely described by Paula, all that data gets transferred onto our bioinformatics pipeline, which is where that data is deciphered into a format that can be interpreted by humans, like clinical scientists. So I'm going to show you a bit of that processing. So once the data is finished bioinformatics analysis, that data gets transferred to clinical scientists, like myself, to look at the changes in the patient's DNA. So now we're in the dry laboratory. This is where analysis of the sequence data takes place. So here, all the data that has been sequenced on the sequencing machine, as seen earlier, gets populated onto a run tracker. And this helps us keep track of which sample was tested on which run, and also guides the scientist on which cases need analysis. We use a traffic light system where red means the data has not been checked at all, and amber or orange, which means it's had one check, and green, which means the sample has been fully analysed and reported. I'm just going to show you what that really means. So here we have a number that is waiting for final authorization by a clinical scientist. So I take the number and I put it on our data system. And here we have this patient, and this is where we view the results. This is a patient, this is a case, a colorectal case, colorectal cancer case. That case has been referred to us to determine treatment options. And we need to know the changes in the patient's DNA, the mistakes that have been made in the patient's DNA that is driving the cancer, in order to know how to target those mistakes. Looking at this data, the system is telling me that this case has a mistake in its BRAF gene, which means this error is what's driving the cancer. And what we want to do is target the BRAF gene with specific targeted therapy. So, to further investigate the mistake, I am going to actually then go and look at the letters of the DNA and see where the mistake has occurred. Here is the sample. So, as Paula mentioned earlier, DNA consists of 4 letters, and the sequencing machine shows us the order of those letters, as you can see. Here, the normal sequence of letters for the BRAF gene in the region we're looking at is GAG, ATT, TT, CACT. Can you spot the error? I'll show you. There. A mistake has been made. Instead of an A, there's now a T. This is the BRAF V600E mutation. Very common in colorectal cancer and highly targetable. So now that we've spotted the mistake for this patient, we go onto our reporting system and enter the results for this patient. And once we're done, we sign it out and it goes to the clinician who uses this information to manage the patient's cancer.